morning, everyone. My name is Kathryn Greiner-Ferris. I am a graduate student at the University of Central Florida, and today I will be presenting part of my dissertation on the population structure of three co-distributed cygnathid species in Florida's estuarine and coastal habitats. So cygnathidés consist of sea dragons, seahorses, and pipefish, and they are found in a variety of marine to freshwater habitats in both tropical and temperate coastal waters found all around the world. And they are unique because, unlike many marine species, cygnathidés lack a larval dispersal phase due to their unique reproductive behavior. So this involves females transferring their eggs to a male's specialized pouches, where they are then fertilized and will incubate for roughly 2 weeks. And then developing youngs are released fully developed and will typically settle into nearby benthic habitats. And in general, cygnathidés and pipefish in particular are considered weak swimmers. So they will typically exhibit restricted mobility as adults. And due to their restricted mobility or limited mobility, dispersal between habitat patches is mainly mediated by rafting. So marine organisms with long-distance dispersal of pelagic larvae often exhibit high gene flow and genetic connectivity across broad geographic areas, which would result in low levels of genetic structuring. So this is different from what we would see in cygnathidés. So species that are direct developers or those with restricted mobility can exhibit pronounced genetic structuring over these small spatial scales. So I'm going to be presenting about Florida. So there are at least 12 recognized species that inhabit the coastal areas of Florida, but today I will solely be focusing on 3 of the most abundant: Cygnathus scavelli, Cygnathus floridae, and Cygnathus louisianae. All 3 species occupy similar home ranges along the coast of Florida, but they differ in their relative abundance and also their habitat preference. For example, scavelli is the only euryhaline species, and they have a greater affinity for shallow benthic habitats and have not been recorded in depths greater than 6 meters. Compared to the other 2 species, floridae prefers greater seagrass coverage. In fact, they are nearly absent from the east coast of Florida due to low seagrass coverage, creating an unsuitable habitat. And in addition to inhabiting coastal marine systems, Louisianae is also frequently found within pelagic sargassum mats. So a previous study by Flanagan et al. Looked at the fine-scale population structure of scavelli in Florida using DD-RADseq, and they sampled from 9 different populations spread across Florida, with samples ranging from 27 to 47 for each population. And their results found 3 distinct populations. The West Coast was genetically distinct from the East Coast, and also a North Florida freshwater population. And populations of floridae and louisianae have also been examined, but to a lesser extent. Mobley et al. Looked at floridae locations using both mitochondrial DNA analysis and microsatellites. For the mitochondrial DNA analysis, they used fragments of the site B gene in the mitochondrial control region, and they looked at all the locations shown on the map, including the white and the red dots. However, for the microsatellite analysis, they only looked at the white dots, which of note did not include Florida Keys, which is the southernmost population of Florida. However, for both data sets, they found a similar pattern of significant structuring between ocean basins. But note that the Atlantic Basin, the way they looked at that was just looking at the furthest populations from Florida, as you would possibly get, North Carolina and Virginia. And then finally, Bertola et al. 2020 used RAD sequencing to examine gene flow directionality of floridae and louisianae, and they used sample sizes ranging from only 1 to 6. So their results showed higher levels of gene flow between locations of louisianae compared to floridae, and they also found that the Florida Keys population of floridae showed pronounced divergence from the other Florida populations, which was a result not found by Mobley et al. However, the authors, due to their small sample size, they couldn't rule out the possibility of these samples not actually being floridae and really being that of a cryptic species. So while individually each of these studies provides valuable insights, the variability between them raises the question of whether these data sets allow for direct comparison of population structure between species. So we can't confidently rule out that the patterns we're seeing aren't influenced by small sample sizes used, or a variation in the number of locations used between the studies, or the fact that they use different sequencing methods. So the objective of this study was to examine the genetic variation of 3 different pipefish species in order to determine if these congenerics show similar patterns of population structure. We co-sampled from all 3 species from locations spread across Florida, and we also increased sample size per location and used a single sequencing method in order to achieve the most reliable direct comparison across species. So to achieve this, we extracted DNA from pipefish samples that were caught by FWC and NOAA as part of their routine sampling effort. And we chose sample locations that would leave us with at least 10 individuals after sequencing and filtration. So for example, for Louisiana, in the one farthest left, that included breaking up the East Coast into North Florida and South Florida. And then also note that the East Coast for the Florida samples is completely missing samples, and that's due to the break in their distribution that was cited by the other studies. And then additionally, our sampling effort for Scavelli also included Biscayne Bay, which was a location not examined by Flanagan et al. So for the sake of time, I'll be brief about my methods, but libraries were prepared for double digest restriction site associated sequencing, and then our data set was cleaned in order to resolve only true single nucleotide polymorphisms using various filtering parameters. And then we used 3 different methods to characterize population structure. This included Bayesian clustering approach implemented by Struck, the program structure, K-means clustering with DAPC, and FST analysis. We also used Mantel tests to evaluate the correlation between FST and distance between sampling locations. So after sequencing, our effort resulted with an average of 4 million reads per sample, and after filtering using the parameters from the previous slide, we were left with 4,192 loci for Scavelli, 5,391 for Florida, and 6,703 for Louisiana. So I'll start with the results of Scavelli. So using the K-means approach by Adigenet, we identified 4 clusters. And then the cluster data set was then visualized by running a DAPC. And the clusters separated North Florida, the entire Indian River Lagoon, Biscayne Bay, and then the entire West Coast. Our analysis by structure revealed slightly different results. So delta K was similar when K equaled 2 or K equaled 3, which indicated 2 potentially optimal number of populations. In both scenarios, the program structure did not distinguish the North Florida population, but the lack of gene flow between the East Coast and the West Coast was very apparent in both. And then apart from the presence of the Biscayne Bay population that was not looked at by previous studies, this genetic break between coasts is consistent with the results by Flanagan et al. Global FST was 0.20 for Scavelli, and pairwise values range from 0 to 0.399. And then our Mantel test revealed a strong linear relationship between FST and distance, indicating that population differentiation in part was due to isolation by distance. For Florida, K-means approach identified 2 as the optimal number of populations, which consisted of Florida Bay with some overlap into Charlotte Harbor, and then the rest of the West Coast. So this is consistent with the analysis by structure as well, which also found 2 to be the optimal number of populations. And then our results are consistent with the findings by Bertola et al. However, remember that these authors hypothesized that the Florida Keys population was likely that of a cryptic species and not actually Floridae. And this was due to the large divergence from the other populations that they observed. However, with our results in the greater sample size and the FST results, our results refute this and suggest that there is a notable genetic differentiation between Florida Bay and the rest of the Gulf Coast. And global FST was lower than Scavelli at 0.02 and ranged from 0 to 0.059, and the Mantel test showed significant isolation by distance with a greater R-squared value of 0.79. So for Louisiana, the K-means analysis resulted in 3 potential populations, but with apparent admixture between coasts. K2 and K3 were determined to be the most likely number of populations by structure, and when K equaled 3, the structure plot suggested very similar clustering to the DAPC plot. And although they used a smaller sample size, Bertola et al. Also found that populations on the east side of the Florida Peninsula were only somewhat differentiated from the populations west of the Florida Peninsula and did not indicate a clear population structure either. Louisiana had the least amount of genetic differentiation between locations, and global FST was 0.01, and pairwise FST values ranged from 0 to 0.032. And unlike the other two species, FST did not increase with distance, and instead remained fairly consistent between the two locations. So this confirmed this was confirmed by the Mantel test, which showed that the mean pairwise FST values were not significantly correlated with geographic distance. So, with all this in mind, do these congenerics show similar patterns of population structure? Well, Scavelli showed the most complex population structure with higher genetic differentiation and more genetic clusters than the other two species. And because they inhabit these freshwater, shallow, nearshore habitats, they are likely protected from the influence of major ocean currents. And therefore, their potential for raft-driven connectivity between populations is much lower compared to the other two species. So their population may exist simply due to geographic barriers to dispersal, which has led to an overall isolation by distance pattern. Although the overall population structure of Floridae also seemed to be shaped by isolation by distance, we also found notable differentiation between Florida Bay, with Charlotte Harbor potentially being kind of like a stepping stone population to the rest of the West Coast. So on the Gulf Coast with no clear barriers, these results suggest that the differentiation that we're seeing here in part might be shaped by local adaptation. This could be due to factors such as temperature, we know the Florida Keys is warming, salinity, or seagrass coverage. And then finally, Louisiana did not exhibit clear population structure and instead revealed admixture between coasts. So remember that this species is one of the most abundant fish species that is found living within pelagic sargassum mats. Which I gave away the ending. This species is one of the most abundant species not found, is found pelagic sargassum mats, which could serve as a mechanism for long distance dispersal, which would allow this typically low mobility species to achieve long distance transport via the Gulf Stream and would promote an overall eastward gene flow, explaining the population structure that we saw. So with that, I'd like to thank all the folks that helped me collect all these samples. I haven't had to go out into the field yet. And also I'd like to thank my lab, the Molecular Ecology Evolution Gaither Lab at UCF, and funding sources. And I'd be happy to take some questions. Thank you. Yep.
I think if I recall correctly, Florida and Louisiana are relatively closely related to each other as species.
Yeah, they're all very closely related to each other.
I'm just wondering whether it wasn't the objective of your study.
Yeah.
Whether you did any analysis to see whether there was evidence of integration among the species.
I haven't looked into that, but I do know some folks who are, I'm going to be sending them some samples to see if there is something like that going on, which would explain, I mean, I definitely ran into not being able to identify sometimes and having to throw some of the samples out. Okay, thank you.
Thank you. Hello, everybody. My name is Joaquin Nunez. I am an assistant professor at the University of Vermont. I'm very excited to share some of our results today. So I want to start by recognizing the work and the efforts of many folks that have contributed to this work. Of course, the members of my lab who presented posters yesterday, and they're right over here. But a lot of the results that I'll share today are actually the sort of the output of a very collaborative effort with members of the European Drosophila Population Genomics Consortium. In fact, I want to highlight that the results of this presentation are only a very small part of a much larger project that should be published sometime in the next couple of months, and this work will actually represent the second release of our resource, the Drosophila Evolution Over Space and Time, so do be on the lookout for that. Okay, So a lot of the work that we do in our laboratory is motivated by a fundamental desire to understand the forces that generate and shape genetic variation in ecosystems that are known for being highly fluctuating, right? In particular, we capitalize on the biology of natural populations of Drosophila melanogaster to achieve this goal. Now, we do this for a variety of reasons. There is, of course, very beautiful biology that these flies allow us to access. They naturally inhabit environments that are highly variable, so we can think of seasonal orchards or seasonal compost piles. And then, of course, as a model organism, they have a very rich literature that we can tap into to contextualize some of our findings. Now, on this last point of the literature, I would argue that there are two general sets of literature related to the theme of seasonality and rapid adaptation in Drosophila. On one side of the equation, which is the one that folks may be more familiar with, is this idea of seasonal adaptation. And generally speaking, the argument is that there is evidence to suggest that this multivoltine organism experiences rapid bursts of adaptive evolution, tracking the changes or sort of the ecological changes as the seasons progress in a process that we call adaptive tracking, driven by a fundamental sort of trade-off between reproductive output and somatic maintenance. And there's an extensive literature exploring that beautiful biology. On the other side of the equation, we have something which is equally wonderful, but perhaps less studied. And this is the idea of seasonal demography. Now, seasonal demography is a very peculiar thing because it actually happens at the same time as seasonal adaptation. So as the seasons progress, it's not just that these flies are rapidly adapting to the changes in the environment. Fundamentally, the demographic properties of these populations are also changing. And to think about this, I want you to imagine, the seasonal orchards in the middle of the fall or the summer where there's sort of plenty of food available because of the harvest. So populations get to have really large, population sizes. But then as soon as those harvests are over and sort of winter rolls around, these populations experience quite severe bottlenecks. And whoever survives those bottlenecks diapauses and then overwinters to start the cycle in the next generation. So a paper that was recently published by Alan Berglund and myself actually looked into some of this phenomena, and we were able to show, using data from one population in Virginia, which we studied extensively, that these overwintering bottlenecks have tremendous consequences for the genetic structure of these natural populations, and part of that is driven by some of our estimates, which show that these bottlenecks can be on the order of 80 or 90%, so quite strong demographic events. Naturally, however, the thing of working with Drosophila melanogaster is that this is a true cosmopolitan species. So we can find populations of these organisms across many habitats around the world. So the natural follow-up question to our work in Virginia is, if we pick populations from different places and we do the type of analysis that we've done in Virginia, will we observe the same type of genomic signatures of overwinter? Would they show sort of natural clinal patterns vis-a-vis temperature, and also whether or not we could sort of tap into some data sets from NASA and from NOAA to understand what aspects of weather may be more or less important to make these predictions. General hypothesis being that the strength of winter is going to play a fundamental role in this phenomena. As I alluded to earlier, the data set that we're using to tackle these questions is the Drosophila evolution over space and time. It's a public resource and is one of the most beautiful things that I've ever worked with. We have samples across many places around the planet, and those samples actually were sequenced multiple times within a year and then across multiple years. So it's truly a great resource to tackle this question. I want to tell you a little bit about the statistic that we've been using to characterize this overwintering dynamics. In a very oversimplified way, we're basically using an application of the fixation index of the FST. And basically what this statistic is doing is taking into, sort of capitalizing on the natural sampling structure of the data. Because, multivoltine organism, which we sample multiple times within a year, gives us a sense of the change of genetic differentiation within a growing season, for year 1 and year 2, for example. But then naturally, if we also do these comparisons across years, our statistic will eventually capture this overwintering boundary. And in fact, some work that we did in this paper that I mentioned earlier sort of explored the idea and provided evidence that there is in fact a correlation or there is sort of power to determine that depending on this sort of year-to-year FST statistic, we can learn something about the strength of this overwintering bottlenecks, right? So I'm going to explore that in the test data set. So this map sort of showcases the effort of that analysis. We selected some samples from the test data set, and then we applied the statistics. In total, we did about 31,000 pairwise comparisons. And then this map sort of showcases the first interesting finding. So each one of the dots is a population, and the shade of green represents the mean value for this year-to-year FST. And here we see the first sort of piece of evidence that is consistent with our hypothesis, because as we can see, the populations that live near the sort of northern range of Drosophila, near the Arctic Circle, in these places that have quite strong winters, do have much higher levels of year-to-year FST relative to other places, yet we do find evidence of overwintering all throughout the seasonal habitat. Another interesting way to visualize this data is just to plot it as a function of latitude. And this is how that data looks like. We can appreciate that there is a correlation that exists between latitude and year-to-year FST. That correlation is quite robust to permutations, which is always great to see. We see those high FST values at the higher latitudes. However, there is something else here that jumps to people's attention. And that is, of course, this quite unexpected tower of outlier year-to-year FST values at a lower latitude. And that is not quite expected. Well, we did a little bit of sleuthing. Turns out that the vast majority of those samples come from one place and 1 year. 2021, Turkey. And we did a little bit of Googling, and it turns out that those samples happen to capture, almost by accident, one of the most significant heat waves in the history of Southern Europe, which in Turkey happened to drive tremendous wildfires which destroy habitats. So it appears that we have inadvertently captured the impact, at least the demographic impacts. We haven't quite done the analysis for any adaptive consequences for that, but we're working on that. But at least on the demographic side, we now have microevolutionary insight into the consequences of these extreme weather events. So that's very cool, but also very scary. Okay, in the last minute or so of the talk, I do want to tell you about our second ambition, which is trying to find ecological variables that could help us predict, perhaps, the dynamics of this overwinter. And to do that, we mined publicly available data from NASA, particularly from the Power Project, another great publicly available resource. Now, the general gist of the analysis that we did is that we try to assess correlations that exist between our statistic, our FST statistic, and a number of sort of ecological estimates calculated between samples, right? So we calculate pairwise FST, and then we calculate ecological sort of summary statistics that cover the same period. Here I'm showing you maximum, average, and minimum temperature, variance of temperature, and also the number of days below 5 degrees Celsius. Variance of temperature is a proxy for the length of the seasons, and the number of days below 5 degrees Celsius is a proxy for the strength and the harshness of winter, right? So longer winters. The red dots are the correlation estimates, and then the violin plots are the permutation analysis. Okay, now here's the table that captures some of the essence of the analysis. So interestingly, aside from minimum temperature, which was weakly associated with the analysis, so it was not really interesting, every other variable that we looked at had a significant association. And the direction of that association was also consistent with the expectation of the hypothesis. So basically, sites that have higher maximum temperature or higher temperature on average, so these are basically sites with really nice warm temperatures, or sites with higher variance, which is to say places in which summer, winter, and fall are much longer, those sites have a negative correlation with FST. So the strength of the bottleneck, as our statistics predict, is much smaller. On the other side, sites that have a large number of days below 5 degrees, so these are places with harsh winters and longer winters, those have quite a positive correlation with our statistics, hence providing another angle of positive sort of validation to the hypothesis, which suggests that places with harsher winters, longer winters, do experience stronger bottlenecks. Okay, so to wrap up some of the results of our analysis, the first take-home message is actually this idea of appreciating temporal structure. right? I think that as population geneticists, we're always very aware of spatial structure as being an important sort of source that modifies genetic variation, but we'll make the case that temporal structure is also a very important thing that we should be looking into to contextualize patterns of genetic variation. More particularly in our story of overwintering, we've actually are now able to appreciate the contribution of different variables to this issue of temporal structure, and particularly the length and the harshness of winter appear to be important players. And then finally, and this is sort of, once again, sort of appreciating the beauty of the test data set, which is a sort of collaborative resource, it seems that our data set has now grown to a point where we're beginning to capture some of this really interesting, or the macroevolutionary impacts of really interesting ecological events related to climate change and extreme events, using the model organism Drosophila melanogaster, which sort of opens the possibility for many interesting further studies. Okay, before my time runs out, I do have a quick plug-in, which is my collaborators, Katie Lauderhus and Nick Teets and we recently got a grant to sort of do this same type of work in a different organism, Drosophila susukii. And we are recruiting PhDs and postdocs, and my lab's also recruiting PhD to continue doing some work in melanogaster. So if you are interested, or anybody you know is interested, please reach out to us. Thank you, folks. Happy to take any questions. Yes?
I was wondering if you had any hypothesis as to why the variance was negatively correlated with FSD.
Yeah, when we look at the data, I think that what the variance parameter is trying to tell us is that places in which spring, summer, and fall are much longer periods tend to have higher variance. So Italy, for example, have very short winters relative to the length of spring, the length of summer, the length of fall. Sweden, on the other hand, has a very long winter. So the variance in that sense is telling you something about the length of the winter relative to the other seasons. So, Spain, for example, has a very nice set of ecological variables. Our flies like Spain. They don't appear to bottleneck too much in Spain. So I think that's what that data is telling you.
So when the variance is lower, the FSD tends to be higher, I guess?
In places where the winter season is shorter, the year-to-year FSD is smaller, which means the bottleneck is less aggressive. That's the argument. Let's go over there.
Will they overwinter now?
We don't really know. There is a lot of hypotheses about compost piles, about leaf litter, about all different things, but we haven't sort of nailed the best possible candidate. So that's still an open question. Yes, sir, in the back.
Great talk. I was curious if you saw evidence of fixation in areas where there's longer winter and a little loss, basically.
That's interesting. We actually don't see any of that. Yeah, and we can talk about this, but there are multiple reasons why we wouldn't expect to see fixation, but we haven't, yes. Ma'am, yeah?
I was wondering if this FSD statistic that you use, is it only capturing the bottlenecks or also the adaptive tracking? And can you somehow
That's a great question. So the way that we've used this FST is as part of a large battery of statistics that capture population structure. We actually use that statistic to then fit sort of Bayesian analysis to actually estimate the size of that. And just very quickly, is the ratio of FST within a year relative to across years that captures in a really interesting way how much the populations are collapsing. So, but yes, great question. Thank you, everybody.